My article was the undetectable intracellular free copper, the requirement for a copper chaperone for superoxide dismutase. Authors are T.D. Ray, P.J. Schmidt, R.I. Pufau, V.C. Colata, and T.B. O'Halloran. So, the importance of this experiment is, and what they're trying to find out, is how cells handle toxic metals in the body, like copper, for example, in this experiment, and how copper chaperones manage these levels in their cells to activate important enzymes like SOD1 specific to this experiment. So there's three main points: understanding the role in our health, since copper is essential but can be toxic if it's not managed correctly in our body, just like everything else. Second one is revealing the mechanism of how copper activates SOD1. So there are proteins, also CCS, which is a copper chaperone that helps copper into our enzymes to function properly. And third are the implications of this process and how it's linked to neurological diseases. So the thesis in this experiment is that the copper chaperone CCS is essential for activating the enzyme SOD1 in vivo by directly delivering copper ions, thereby bypassing the intracellular copper scavengers that collect copper ions and risk the possibility of interfering with essential copper-dependent enzymes. You can see in the picture, there is copper. Here it goes collected by copper chaperone and directly entered into the enzyme. So the problem, and what we're trying to figure out, is understand how the cell safely and efficiently uses the copper for enzyme activation, even in a low copper environment. And the hypothesis that we had is the copper chaperone is essential for activating SOD1 in the body by delivering copper directly to SOD1 in environments where free copper are extremely limited. So there's 4 data collection points here. There's biochemical assays, genetic manipulation, elemental analysis, and polyacrylamide gel electrophoresis and protein staining. So biochemical assays, we use this to determine how much of the SOD1 activity there is and see how much there is with or without the copper. Genetic manipulation, in the experiment, we use modified yeast strains to represent the human cells to get an in vivo experience for the experiment. Elemental analysis to measure the copper levels to ensure that the copper content is the same, so there aren't variations which will cause error in the experiment. Polyacrylamide gel electrophoresis protein staining. This was used to visualize how active the SOD1 and just to confirm that there was presence of copper in the SOD1. So the first thing here, Figure 1 Panel A, what first we did was trying to purify the CCS copper chaperone from E. coli bacteria. This experiment was just a kind of a pre-lab to prepare for the actual lab and determine if our hypothesis was right. Panel B, this was trying to figure out if the CCS was necessary for the activation of the enzyme. So there were two things added: the CCS and GSH. So this was just to see if the CCS was necessary for the activation of SOD and see how well it worked. And so first there was a BCS used, which this helps this attaches to the free ions and mimics the low free copper environment. And then the CCS and GCH were added independently. And we can see in the top right here that with the CCS, there was the most SOD activation because it was directly inserting the copper into the enzyme, while everything else, it was not as low. With the CCS and without the BCS, there was almost just as much activity. So CCS here is definitely crucial for the activation of the enzyme. And panel C, This is trying to determine if the activity at the SOD1. So the presence, just trying to determine how activated the SOD1 gets. So SOD was very, as you can see here, SOD1 was activated with the CCS, and even in the presence of the BCS, but without the CCS, there was nothing delivering to the enzyme, which caused no activation. So what we can take is that CCS is essential for activating SOD1 environments, even in low copper. So as we can see in the other experiments, it was pretty obvious that there was more activation of the SOD1 enzyme with a copper chaperone. In figure 2 and panel A, we're trying to test we're doing this in vivo now, so with the yeast cells. So this is now in a real life cell that was genetically modified to represent a human cell, and we can see that with the CCS, there was, under all conditions, there was SOD1 activity, but without CCS, there was no activation in low copper levels. So this shows us that CCS is required for enzyme activation in low copper levels, which is more like our body. Our bodies have very low copper levels because copper can be toxic in our bodies. The next, in figure 2, panel B, we can see that we're now trying to see if metallethionines are required for the activation of SOD1. So what a metallethine is, it pretty much attaches to copper and allows the CCS to transport the copper. And so we can see that with the strain lacking the metallethine, but it's not very much SOD1 activity. But with it, there's a lot more activity, and it indicates that there needs to be metallethionine inside of our cells for the copper availability within the enzyme. And the conclusions is that CCS is crucial, obviously, and especially in low free copper conditions, and that metallethionine plays a very key role inside of the giving free copper to the copper chaperone and then eventually into the SOD1 proteins. So the limitations of the experiment are that we can only get to the conditions so close to human cells, and so like the yeast cells, there aren't exactly like human cells, and that the copper amounts are so low inside of these cells that we may not be able to well measure them. So, for example, like the amounts of copper in like the yeast cells may have been so low. I mean, they have proper equipment, but like it's so low that the numbers might be off, which will lead to error. So the conclusion of this experiment is that CCS is required and is very essential, especially in low copper environments, and that metallethine is very important to make sure that the copper is movable for the copper chaperone, and that these enzymes are important to keep our cells from reaching toxic levels of free copper ions. This is the reference. I just used the article.